Okay, thank you so much. So, before anything else, class, I would like to ask of you, so you can chat your answers. How are you feeling today? So, how's your morning? So, in just one word, please type your answers here in the chat box so that I can read your answer. How are you feeling right now? Oh, Jan said she is fine. Also, Jasmine is also fine. Krista is feeling great. Wow, and also Krista is also good, very good. So you have a good morning. It's really real that you are really feeling fine and you're okay. So I'm also glad that you are safe and sound there at home. So while we are still continuing this online class, okay? So allow me to share my screen right now. And mhm. So, class, this is your InBt152 molecular toxicology laboratory. So, before anything else, class, so that we will start the morning actively. So, we will play a game right now. It's called Four Picks, One Word. Okay. So you can just do not type your answers in the chat box because I cannot read it anymore because I'm sharing my screen. Okay, so you just have to answer right, using your microphone right away. Okay, what's the answer of this four picks, the word? Okay, so understand, class?
Yes,
Yes,
ma'am.
ma'am.
Okay, so let us start now. So what is this? Okay, it has four peaks. So what's the word? Anyone?
S- Ma'am,
Slow,
slow.
slow.
Very good. Yes, that is slow. That's the answer. Okay, the snail and also the ant. Second picture. How about this?
Beat,
Come again.
beat.
Beat. Oh, yes, correct. That's beat. The right answer is beat. It's a heartbeat and the sound of beat. How about this?
Volcano,
Volcano, ma'am.
Yeah, that's right. Oh,
Volcano, ma'am.
so intelligent. That's volcano. I
volcano.
guess you are playing this in your cell phone, no? So that this is a famous game app actually. So I think it's yeah. How about this one? The
Cold,
second to the last.
Cold.
Cold.
Coal. Yeah. Coal. Yeah. Very easy. Okay. The last one. How about this? What's the word?
dream,
Dream,
Yes.
ma'am.
Dream. Correct. Yes. So you're so, ano na, you're so good at this, no? So I guess your brain is already active and it's already have moved on already from sleepiness. So we'll now start with our proper discussion. Okay. So as a review class, okay, we have already tackled about three exercises, and exercise one is entitled "The Finding Data on Compounds in the Chemical Literature." Okay, it's all about the material safety data sheets. Okay, who can recite or who can answer what's the importance of material safety data sheet? So, who can answer, class? What's the importance of material safety data sheet?
Material safety data sheets, ma'am, allow the downstream users to be able to work safely and be warned with the possible chemical hazards that is associated with a particular chemical.
Very good. That's right. So material safety data sheet, it provides information about the chemical structure, about the identification of the chemicals, and also the toxicological hazards in order to prevent mishandling, in order to prevent accidents for the handlers and also the other persons and also the environment as well, okay, for protection from the environment. Okay, the exercise two is entitled Finding Appropriate Reference Sources of Toxicological Hazards of Chemical Substances. And the exercise three, which is the last exercise, which I required all of you to conduct this at home because the materials are available there at home. It's just so simple and it is entitled Determining Threshold Limits, the Test for Salt Solution. Who among you here have already conducted this experiment?
Ako, ma'am.
Muriel, oh, you have already conducted. So how was the result of your experiment?
Not yet, ma'am. I just started yesterday, ma'am.
Okay, so you have already gathered the result. Have you already let your household member of your household tasted the salt, the different concentrations of salt already?
Yes, ma'am.
Okay, very good. So thank you so much. Let us now proceed to another exercise, which is entitled the hydrogen cyanide analysis on roots. Okay, so it's all about the cassava roots, the hydrogen cyanide on cassava roots. Okay, at the end of this discussion, so you must be able to learn the technique of rapid evaluation of cyanide content of cassava roots. And the last one is to determine the hydrogen cyanide content of different parts of cassava roots. Okay, so, who among you here missed this cassava-based food snack with 1,000 vitamins by Borge and BSU? So, um,
Oh, ma'am.
are you fond in buying this food snack during the afternoon?
Sometimes, ma'am.
Sometimes very good. Yeah. So, si Borge has already uh, Borges is always claiming that his snack or the lead kid has 1,000 vitamins. No, it is a cassava-based food snack. So until now, Borges is still selling this one, pero naanak siya, it's outside the gate. So I'm still buying this lead kid. So it's still delicious. Okay, so I have asked you this because this snack or the ligid is made of a cassava. Okay, so there are also other products that are made out of cassava or cassava grapes, cassava tubers. So this another product is the cassava kitty that is available in the Philo cups. So the Philo cups are producing products, cassava-based food products in order to sell it. So that's the importance of the cassava right now, no? So cassava or the Manihot esculenta L is a woody shrub extensively cultivated as an annual crop in tropical and subtropical regions for its edible, starchy, enlarged roots. Okay, so indeed it has lots of vitamins, but it's not necessarily 1,000 vitamins, but it has lots of nutrients. It is rich in vitamin C and potassium, repair and growth for our body tissue, rich in iron and magnesium, which helps for the production of red blood cells and supports proper growth. And also it has magnesium to keep our nerves and muscles function as well. It is low in saturated fats, cholesterol, and sodium. So this cassava class, the cassava root is um, known to be a poor man's crop. Actually, it is the third largest source of food of carbohydrates in the tropics after rice and maize. So a lot of people in the province planted this kind of crops because it can really sustain the nutrition needed for our body. So it is a major staple food in the developing world. It provides a basic diet for over a half billion people. So with this, the plantation or the production of this kind of cassava is friendly to most to drought. So it is the most drought tolerant crops that is capable of growing on marginal soil. So here in Leyte, there are many lands that are utilized for the cassava production. And actually in Philippines, it has lots of germplasm for different varieties of cassava. Okay, so these are the cassava-based food products that are processed that are also available here in the university, particularly in Philippines, and also other cooperatives in other barangays that are also utilizing this one. Okay. So, however, the demand of this cassava product has become limited because of this threat, because of this content that are in the cassava tubers. So in the last year, in the last 2005, there was an incident reported that there was a food poisoning that kills 27 in the Philippines because they ate cassava. So particularly it was in Bohol. So there was an incident that time that they ate the cassava-based food snacks, and indeed majority of them suffered from food poisoning, and also others, there are two, I guess, that have died. So these are children. So the cassava has really this compound that is really threatening, that can be an agent for that is can be poison to humans, can poison to humans. Okay, so that is why, and this compound is called the hydrogen cyanide. Okay, so the hydrogen cyanide is called the hydrocyanic acid or the prussic acid. It is extremely poisonous. It's a colorless liquid with a bitter almond odor. Now, this is the chemical formula of this compound, the HCN, and the term cyanide is referring to the functional group. Okay, the carbon with three covalent bonds with the nitrogen element. Okay, so where does this, where it can be found? So it can be obtained from fruits that have a peat. Also, it is available in, it is also can be found in cherries, apricots, apples, and bitter almonds. But these peats contain only small amounts of cyanohydrin or hydrogen cyanide. This also can be found, this compound can also be found in exhaust of vehicles, in tobacco and wood smoke, and burning nitrogen containing plastics. So the bitter roots of the cassava plant may contain up to 1 gram of hydrogen cyanide per kilogram. So that is why this cassava is really processed well in order that this hydrogen cyanide will reduce. Okay. Okay. So the specific compound that is toxic in the cassava is called the linamarin. So this is the chemical structure. Okay. So it has two different cyanogenic glucosides, the linamarin and lutastraline. So the linamarin itself produces toxic compound hydrogen cyanide, which is hazardous to the consumer. So Class, this linamarin, how does this converted to hydrogen cyanide class? So the linamarin in the cassava is active, is hydrolyzed by linamarase enzyme that is present also in the cassava. Okay, so then this linamarin is hydrolyzed and then be converted into free hydrogen cyanide. So this free hydrogen cyanide is toxic in higher animals. So this hydrogen cyanide or the HCN is will combine with the cofactor which is present in the hemoglobin. That is, it will bind to the iron 2 plus, which accounts for the formation of cyanohemoglobin, and that will result to food poisoning. Okay, so another mechanism that HCN or the hydrogen cyanide also due to molecular mechanically, it also interferes with the oxidative phosphorylation pathway by combining with cytochrome oxidase and inhibiting electronic transportation. So it disrupts the ATP cycle or the ETC cycle in order to prevent the formation of ATP. Okay, so that's it. So a few milligrams of the substance and of related cyanides can be rapidly fatal to humans. So with this, it's acting by blocking the ability of cells to use oxygen. But because this hydrogen cyanide binds to the ferrous to the iron 2 plus, so it prevents the binding of oxygen to the hemoglobin. So because na may bind nila which is the HCN, so the CN binds to the Fe2 plus, so the oxygen will not also bind to the F2 plus ions, and it will prevent the distribution of oxygen throughout the body. Okay, the amount of the cyanogenic glucosides varies depending on the cultivar and growing conditions. So, plus not all cassava tubers are really high in this HCN, but it varies in different varieties and also different growing conditions. So that is why it is important for this that the HCN has to be determined its concentration. So these are just, I will just run down to you the symptoms of acute cyanide intoxication. So these are rapid respiration, drop in blood pressure, rapid pulse, and also the dizziness, the headache, the stomach pains, vomiting. They will also experience diarrhea, mental confusion, twitching, and convulsion. Okay, so due to the increasing demand of this cassava, many methods were used to improve the palatability of conversion of cassava root into stable products. So they have employed many processing techniques that includes peeling, soaking, boiling, fermenting, and drying. So with the poor processing of cassava, it results to cyanide poisoning and poor quality of cassava's final product. So it has really a strict protocol when you use this cassava grapes. So you have to peel it, soak it, and boil it. So that's really a must to do in processing cassava. Of course, yes, in order to prevent cyanide poisoning. So that is why so we have different content, hydrogen cyanide content in different varieties of cassava. So it is really important for us, it, since we are talking about toxic compounds no, in the hydrogen cyanide, it's really important and a must that we should know how to determine the content of this hydrogen cyanide. Okay, there are different methods in which I have to discuss with you two kinds of methods that should be that can be done in the laboratory and also can be done outside the laboratory. Okay, so one, so the cyanide content determination first. This you can use the thermo scientific Orion star or the combination cyanide electrode. So you can use this electrode for cyanide content determination. You just have to calibrate using 1 ppm and 100 ppm standard solution of potassium cyanide added with 1 ml of sodium hydroxide solution. Okay, so actually, there is a one laboratory that is available, which is in the microbe and fermentation laboratory under the provision of supervision of Dr. Julie B. Tan. So along with their pH meter, so they include this cyanide electrode because they are utilizing cassava grades for their food products so that they will know the content, the levels of cyanide that are reduced during their processing. Okay, so third is you just have to dilute. So this is just the rundown of the procedure. Dilute the 10 grams of sample with 90 ml distilled water and mix it with 1 ml sodium hydroxide solution. So it's just very simple that you have to dip the cyanide electrode into the sample. And then after that, you have to read the uh, instruments monitor and you have to calculate the concentration level using this formula. The cyanide level is milligram per kilogram equals to the ISC meter reading that is found or that will be appearing in the monitor divided by the weight of the sample, which is 10 grams, and also which is F is 10 grams times the volume of water used in ml volume. Okay, so yes, the ISC is the reading of this in the monitor. Okay, so that's how simple it is if you are in the laboratory, but you cannot. I don't know if maybe there is a kit or just a rapid kit that can really just you just have to bring it into the farm so that you can really know the cyanide content of it. But yes, it has already updated their instruments from different brands and companies. Okay, so another method that is used for the cyanide content determination in which this is the method that you will discuss it in the lab report class. Okay, so this is called the pipette paper method. So the method gives the potential cyanide content of cassava roots on a fresh weight basis. Okay, so this is just limited for fresh weight basis. For the using the electrode, you can measure the content of the cyanide from a something like a cassava grade, mga processed foods already. Those cassava-based food products now you can really just use the electrode for the determination of the content. Okay, so but this pipette paper method is limited only for fresh weight basis, in which you can really bring this to the field and do a rapid detection or concentration level of the HCN. So it relies on the endogenous enzymes to break down the cyanogenic leukocytes or the linamarin in fresh cassava root samples. So, okay, how does it work? So, class, so the hydrogen cyanide liberates in the process in which it reacts to the picrate papers that have a color pigment, and this reaction will darken and with the release of cyanide. So the darker the color of the picrate paper, the higher the content of HCN. So that's the simple principle of the picrate paper method. Okay, so with this experiment, So this is just the illustration in which you can have a visible picture for this, since we are not doing this in face-to-face right now. We're doing a virtual laboratory class, so that you can have a vivid picture of this. So this is the setup for most of the experiments. So you have this, you can use screw cup, test tube, or beaker, okay, but as long as it has a cup, it has a screw cup, okay. So you have this thick rate paper inside, and it has the 100 milligram of cassava root sample inside the tube, and also the water round paper disc containing buffer at stage 6. And you can use plastic bottle or the screw cup test tube, just like here. Okay, and then this is the plastic that support the pick rate paper. Okay, so you can also use this small containers, okay, as long as there is a cup. Okay, so that's the simple setup of this quick rate paper method, in which you can really do it in the field when you want to rapidly determine the HCN content of different varieties of cassava. Okay, so the variation or the different levels of the darkening of the papers is measured to the amount of hydrogen cyanide released from fresh cassava root samples. So many reports have argued that this method is not that reliable because, of course, it is a colorimetric method and it's just based on color. But it is the best option if you are far from a laboratory. So I have said earlier to you that it is you can really do it in the field. Okay, and the key to here is to focus, to have focus, precision, and timeliness. Okay, so that's the simple method for the HCN determination. So these are the materials class that we have to use: the sodium carbonate, the picric acid, chemicals that we have to use, the Wattman #1 qualitative filter paper, the picric papers, the test tubes, beakers, soloin, and also the plastic cup. So I just have to share with you the results of the experiment that we have conducted when I was still undergraduate, so that we can really have that vivid picture of the results of this experiment. Okay, so yes, this is the color chart and the picture paper with different colors, and it corresponds to different concentration. Okay, so you have also beakers and test tubes that you have to use. So class, this is the procedure of your experiment of this pick rate paper method. So first, you have to prepare alkaline picric solution by dissolving 25 grams anhydrous sodium carbonate and 5 grams moist picric acid in a liter of distilled water. So you just have to calculate the needed amount for a particular volume of the solution that you have to prepare. Okay, if you are preparing 500 ml of this solution, you just have to weigh 12.5 grams of sodium carbonate and also 2.5 grams of picric acid. Just have to calculate, depending on the volume that you have to prepare. Okay, so I guess you have already you are already well and good in this calculation and in preparing for the preparation of the solutions. Okay, so after that class, you have to cut 1 by 6 centimeter strips of Wattman #1 qualitative filter paper. Okay, so you just have to do like that. And then third, you have to dip the filter paper strips into a stock of white crate. Okay, so this what my number one quality paper is a white color form. So when you dip this filter paper strips, it will form a yellow color. Okay, in the stock white crate. Okay, it will have a yellow color. And then after that, after dipping it into the picrate solution, you have to drain from excess liquid before use. And then in the preparation of your cassava roots, of your cassava samples, okay, you have to macerate 1 gram of cassava root in a test tube using a glass rod. Okay, excuse me class, I will just allow others to join. Okay. Okay. So you have to prepare your sample by macerating 1 gram of cassava root in a test tube. Okay. So you should have to know how to should peel the cassava root, and then you have to do it do it appropriately in peeling the cassava roots, the cassava tubers. Then you have to slice 1 gram of cassava roots and then place it on a test tube. Okay, you have to do it using a glass rod. And after that class, you have to add 5 drops of toluene in the test tube, in the cassava sample. Okay, just add 5 drops of toluene. And then after that, you have to suspend immediately the filter paper strips saturated with alkaline citrate above sample by holding in place with plastic cup or rubber stopper. Okay, okay, you have to suspend this filter paper, and then you have to cover the test tube with a rubber stopper or the screw cup itself. Okay, so after that, you have to allow this uh, experiment or the setup to stand at room temperature for 24 hours. So after that, after 24 hours, of course, if we are doing this face to face, we have to go back the following day, and then we have to rate the colors of the filter paper according to their intensity of red color. So this is the color chart, the cassava cyanogen kit color chart. So this is the rating that you have to do. You have to compare it with the sample, the result that you have after 24 hours. So we have these ratings, and then these these are the different concentrations of different colors that are in the color chart. Okay, and this 1 ppm is equal to the 1 milligram hydrogen cyanide per kilogram cassava. Okay, you just have to compare this with the standard color chart. So, and then, so class, this is the setup of this HCN determination using Pickrate method. Okay, so this is the cassava root, the cassava sample. So that was sliced and macerated, and then it was allowed to stand at 24 hours. So this is the color of the paper strips at 0 hour. As you can see, class, that they have the same colors, the yellow color. Okay. So after 24 hours, this is now the result of the um, experiment. Okay. As you can see, class, that we have tested different varieties, the slimy and not slimy variety of cassava. And we have also uh, picked different parts of cassava roots, which is we have this the tip roots, which is in the represented in letter A and B, and also the middle root part, and C and D, and also the rest, the neck part of the root. Okay, so uh, these are now the results of the experiment. It has different colors that we have. And this class, okay, this is the result that you have to discuss in your lab report. Okay, so we have already a data to analyze for this. Okay, we have the rating, and you should have class you have to compare it with the color chart and present the different concentrations of the HCN, okay, in your lab report. Okay, so that's the result of the colors of the different parts of the cassava roots. So, yes, that's it for the exercise 4. Do you have any questions, clarifications before I proceed? Class, are you still there?
Yes, ma'am. None so far, ma'am.
And so, is it clear with the class?
Yes,
Yes, ma'am.
ma'am.
It was
Yes,
clear.
ma'am.
Okay. So there are no questions. There are no questions. So this is just the instructions on how to prepare and submit the laboratory report. Okay. Just have it must consist of the title, the introduction, the objectives, the methodology, the results and discussion, the answers to guide questions, the conclusion and recommendation, and the last is the references. And your laboratory report must be encoded, okay, with a specific font style and size. It's the Times New Roman with a 12 font size in a short size of paper with two spacing between lines. And it should be submitted in a PDF format. Okay, I will put the submission icon there in the VSUEE for the laboratory report. So always, please be reminded, class, that from this time being, you have 3 weeks to accomplish your lab report. Okay, so within 3 weeks, you have to submit your lab reports. Okay, is it clear with the class?
Yes,
Yes,
ma'am.
Yes,
ma'am.
ma'am.
Um,
Ma'am, wala tay guide questions, ma'am. Karon nga lab.
Yes, we have no guide questions because I have provided you this data that is to be discussed in your lab report. I have not included a guide question anymore because meron tayong um, data that has to be discussed by you. Okay?
Okay, ma'am.
Okay, ma'am.
So if there are no questions, so this is just a review of the lesson that I have tackled. Um, these are just the summary questions. Why is it important to measure the HCN content? Who can answer?
In order to be able to determine the possible concentration of kanang poison, ma'am, or kadtong cyanogen, sa cyanide concentration sa cassava, ma'am.
Yes, yes, it is really a must. You're correct that you have to measure the HDN content in the cassava-based food, in the cassava roots, and also the cassava-based food products, so that we'll be aware of these levels that might harm, that could pose a threat to the consumers itself. Okay, so second question, who can answer what is the principle of the pick rate test method for HCN determination? I have already discussed it with you. So who can answer the principle of the pick rate test method?
Ma'am. The darker the color, the higher the concentration, ma'am, of the ano, poison.
Correct. So the hydrogen cyanide will react to the picrate paper, which contains the picrate solution. So, and it forms a color. And the darker the color of the picrate paper, the higher the concentration of the HCN. Okay, so you're correct for that. So, uh, for the announcements class, okay, so I will just inform you that by next week, we will have a quiz for the exercise 3 and 4. Okay, it will be posted in the BSU EE. And please be reminded of your lab reports that was already due for the exercise number 1 and 2. Okay, you have to submit it class already because it's already deadline. Okay, are there any questions or clarifications?
None so far, ma'am.
Okay, I will now. Thank you so much, class, for listening. But class, before I dismiss you, let me share to you, class, this saying that learning is not attained by chance. It must be sought for with ardor and attended with diligence. Okay, so we have to strive hard. We have to persevere in these times of um, pandemic that we are in the online platform of learning. So it is not happened by chance, but it is really has to be worked for. Okay, uh, we have to be persevering in all the times. Okay, so I know that we have lots of requirements to do, but one thing is for sure that you have to really do it with diligence, okay? So that you will not cram in the day of deadlines, ganun. So you just have to do it right away. Be inspired in doing your lab reports and also attending your class. Okay, so thank you so much, class, for listening and thank you for joining the online class meeting. Okay, so before I will dismiss, can I request everyone to open their cameras and let's have a photo opportunity. Okay, how about the others? Krista Abumay and Krista Rosette. Si Krista Rosette. Okay, let's okay, wait lang. Okay, so let's have a photo opportunity. Everyone smile. Okay, wait lang. Ito arong kuan, group chat. Okay, another ano, kuan, another picture. Okay, smile. Okay, thank you kaayo class for attending the meeting. So, kanto sa ba na-dismiss sila ni ma'am? Nakabantay mo?
Kuan, ma'am, after nimo atong last slide nimo, ma'am.
Ah, okay. Tumoto. Sige, salamat kaayo class for attending. Thank you, Mariel,
Thank you,
John,
ma'am.
Ryder, Krista,
Happy
Sister
Teacher's
Rosette.
Day, ma'am.
Thank you so much.
Teacher's Day,
Happy
ma'am.
Teacher's Day,
Thank you,
ma'am.
ma'am.
Thank
Thank
you,
you.
ma'am.
Bye-bye. Magkita na po na next week na po.
Hi,
Okay.
ma'am.
I
Bye,
will just,
ma'am.
bye-bye. God
Bye,
bless. Thank you so
ma'am.
much.
Amping.
Bye-bye. God bless.